to you.
Yes, hi everyone. Thank you, Julian, for a kind introduction. And I just realized a blunder that I have over here. I never really realized that thanks to the pandemic and the remote nature of the talk, I'll be speaking to you. I'll be sitting in a time machine and speaking from the past, but here we are. Yes, so as Julian said, I'm interested in utilizing single-cell genomics to study the human cerebral cortex. And as you've heard from beautiful talks from both of the speakers, the cerebral cortex contains an astonishing diversity of neuronal and glial cell types broadly distributed across 6 layers, although as has been pointed out throughout the presentations today, there are a number of variations in the laminar architecture, depending on which functional area you're looking at. And for example, such that, for example, in the visual cortex, the layer 4 will be split into 3 sublayers. And in other areas, there'll be a slightly different architecture. So really, if you survey and if you compare the laminar architecture across the cerebral cortex and the prospective functional areas that have been surveyed by Santiago Ramon y Cajal, as well as Brodmann and von Economo, there's quite substantial variation across the cortical areas. So one of the fundamental questions in the field is how do we achieve this rich diversity that eventually bestows the ability for the cerebral cortex to perform such complex computations. So importantly, the different areas, the different anatomical and functional areas of the cerebral cortex tend to also differ in terms of their both cellular composition and connectivity patterns that eventually establish this distinct functional properties for these areas. And this is quite interesting because that suggests that there may be differences in the developmental origin and the neurodevelopmental programs that are initiated in these different prospective areas during early development. And so, as you've heard today, the cerebral cortex emerges and the diverse neurons and glia emerge through a series of neurodevelopmental events presented here or illustrated here by a drawing from Conrad Waddington, where a neuroepithelial or radial glia stem cells sort of traverses through different states in development and undergoes consecutive changes and consecutive decisions during development. Now, every now and then, this process can go wrong, and there's a number of devastating neurodevelopmental as well as neuropsychiatric disorders that together are thought to affect somewhere around 1 in 6 children in industrialized countries. And for most of these disorders, there are virtually no treatments. So a quick overview of the human cortical development and forebrain development. The brain emerges in very early development and becomes specified at just a couple of weeks after conception, and it really, the initial formation of the cortical of the neural tube then undergoes evagination and the neuropole closes progressively from this middle part, both in the posterior and anterior neuropore. And after the closure of the neuropore around 3rd week of gestation, the anterior portion of the neuropore of the neural tube undergoes a substantial swelling. And this swelling indicates the future telencephalon, or part of the neural tube, which will later give rise to both the cerebral cortex, but also basal ganglia. And so if you were to take a cross-section through this, you would see a structure, a cross-section that looks like this in the human, where you can see the lateral ventricles, and this is where the swelling occurs. And on the outside of that ventricle, there is the sort of semi-uniform neuroepithelium that differs actually quite substantially depending on which part of the cerebral cortex you're looking at. And so over the years, it's been appreciated that across evolutionary spectrum, there has been quite an expansion and quite a number of changes in the size of the cerebral cortex. But it is important to note that this expansion of the cerebral cortex that is most notable in humans did not happen equally across the functional areas. And so such that the prefrontal cortex, the higher order association areas of the cerebral cortex have expanded disproportionately in humans respective to other primates and even the mouse. There are also a number of differences in neurodevelopmental events that have been recognized through studies using comparative approaches with including lineage tracing, including histology, including marker gene expression analysis. And this schematic over here summarizes sort of a global view of some of the major differences between mouse cortical development and human cortical development, such that the mouse cortical development, which, as you've heard from Song Hai's and Alfredo's talks, undergoes the sequential progressive changes in neuroepithelial, from neuroepithelial stem cells through to radioglia, which then undergo neurogenesis and later also gliogenesis. The human cerebral cortex broadly follows the same sequence of events. However, there are notable distinctions. For example, the radioglia neural stem cell population, which is largely confined to one type in mouse, becomes expanded in its diversity. And the secondary proliferative population, the secondary population of radioglia called outer radioglia, they populate the outer subventricular zone. And in fact, the expanded outer subventricular zone has been for a long time recognized as one of the major differences in the organization between mouse and humans. And so it's been sort of classically thought that the fundamental events that give rise to the neurons of the cerebral cortex, especially excitating neurons that involve differentiation of radioglia via directly into neurons or via intermediate progenitor cells, which are transient amplifying cells, as you've heard about them in earlier talks. It is generally thought that this process is largely uniform across the prospective areas. And so the fundamental question is that we wanted to address is where do the differences occur and how do they emerge? So some hypotheses that have been really fundamental to this have come from work from Denis O'Leary and John Rubinstein and others, and included the identification of patterning transcription factors. These are transcription factors whose expression is induced by local organizers and which largely form these gradients of gene expression across the neuroepithelial sheet. However, this is in contrast, as mentioned in earlier talks, with the sharp and distinct boundaries between functional areas that you can find in the adult. And so one of the fundamental questions that we would love to be able to address is how do you translate this graded expression of patterning transcription factors into a very distinct patterns and with sharp boundaries between functional areas. So some years ago, we have utilized the emerging technologies with single cell transcriptomics. This is a method which you may have heard of. This is a method where you take intact tissue samples, you dissociate them either into single cells or nuclei, you encapsulate these cells in droplets, and in these droplets, there are all the reagents necessary to perform reverse transcription and cDNA amplification. And so by profiling cells from sampled from primary human tissues from prefrontal cortex and visual cortex, we were able to directly compare those lineages in intact samples. So these are from same individuals. And unbiased single cell analysis and data analysis shown here utilizing a UMAP, which is a two-dimensional projection, which groups the cells according to the identity shared gene expression profiles. And so the way this is organized is that down here at the bottom are the progenitor cells, and as you differentiate over here, you start seeing post-mitotic neurons and progressively more and more mature neurons. You can organize the cells in gene expression space according to what's known as the pseudotime. And the pseudotime analysis, the idea behind the pseudotime analysis is sort of like looking at from above at a cross-country race. If you see runners along a cross-country race, you can sort of organize them according to a path, and that gives you an idea of where the cells, the dynamics could be for gene expression changes during differentiation. And so one of the things that was really striking that stood out to us immediately when we saw these types of patterns was that the differentiation into excitatory neurons between PFC and V1 lineages actually really strongly diverges during differentiation. Importantly, these are not just simple, individual markers. In addition to the individual markers that have been long recognized, there are also important or interesting patterns that differentially specify excitatory neurons in either prefrontal cortex or visual cortex, including, for example, the expression of BCL11B and SADB2, which are both regulators of connectivity, such that BCL11B in most areas, in most studies, are enriched in layer 5 neurons, pyramidal tract neurons, whereas SADB2 is a transcriptional regulator which determines the identity of callosal projection neurons. And in primary human tissues, we find these genes to be sort of differentially misexpressed in the visual occipital areas, but co-expressed in the prefrontal cortex. And when we project across the pseudo-time trajectory the expression of known marker genes of intermediate progenitor cells, we can map the time in differentiation when these lineages diverge. And so with transcriptomics, what we are able to determine is that this significant distinction or transcriptional divergence in those lineages occurs post-mitotically, so after the cells have differentiated from intermediate progenitor cells. Of course, this provides a sort of a map of gene expression changes during differentiation into distinct lineages. And what we wanted to also know more about are what could be the regulators of that differentiation. So what is the map of gene regulation underneath the map of transcriptional differences? And to achieve that, we turn to another technique that can be implemented using single cell analysis. And this technique is called the assays for transposase accessibility by sequencing, which was developed by Will Greenleaf's lab at Stanford. And it essentially uses a hyperactive TN5 transposase, which preferentially integrates Illumina barcodes into open chromatin regions of the cell. And when we repeated this experiment on parallel samples, so these were actually samples from the same individuals, we were able to sort of recapitulate the same pattern whereby the progenitor cells tend to group together, but as you follow the differentiation trajectory, you start seeing these lineages diverge very strikingly. Now, interestingly, in contrast to the transcriptomic efforts and the picture in transcriptomics, the divergence between prefrontal cortex and visual cortex lineages occurs actually at the time when EOM's accessibility is the highest. So this is also known as TBR2, which is a marker of these intermediate progenitor cells, suggesting that the chromatin state distinctions during excitatory lineage neurogenesis distinguish neuroprogenitor cells before there are substantial transcriptomic differences between those lineages. So putting the models side by side, we think that there is a remarkable difference in that chromatin state changes sort of foreshadow the emergence of these distinct excitatory neuron lineages in the human cerebral cortex. Importantly to note, transcriptional analysis, differential expression analysis in intermediate progenitor cells does identify subtle differences. So there are genes differentially expressed between PFC and V1 intermediate progenitor cells, but in contrast, the chromatin accessibility, so differentially accessible loci, are substantially more pronounced. There are many more features that distinguish these intermediate progenitor cells in chromatin state space compared with transcriptomics. So together, a model that we hypothesize might be occurring is that the transcription factors that are differentially expressed across the protomap of the cerebral cortex act as sort of pioneering transcription factors to initiate different lineages, differentiation of excitatory neurons into different lineages. This model is congruent with the possibility that there are further changes that might be occurring postmitotically, for example, signals from the thalamus and area-specific neuronal maturation, as well as functional evoked input that could further refine these, what we refer to as proto-regions of the cerebral cortex. And you can read more about this in our recent review. Next, we wanted to see if we can use this information to predict what sort of regulators might be actually playing a role in this differential differentiation of PFC and V1 lineages. And to achieve this, we identified differentially accessible peaks between prefrontal cortex and visual cortex excitatory lineage cells. We next ran transcription factor motif enrichment analysis, and in addition to known transcription factors that have been long established to differentiate those two lineages, we also found some unexpected new candidates, including many members of the retinoic acid receptor signaling pathway transcription factors, which were strongly enriched among the open chromatin regions differentially accessible in the prefrontal cortex, suggesting a hypothesis that perhaps retinoic acid signaling might be playing a role in this differential regulation of excitatory neuron lineage specification. We decided to test this hypothesis using cerebral organoids. Many of you know that pluripotent stem cells can be guided through directed differentiation into neural progenitor cells. And when those cells are cultured in 3 dimensions, they self-organized into these beautiful 3-dimensional structures that resemble some of the early differentiation of the cerebral cortex. And so we exposed these organoids either to a culture media that contained vitamin A, which is of course a precursor to retinoic acid, or we withdrew vitamin A altogether from the media throughout differentiation. We also included a control where we both added vitamin A, but also included a selective inhibitor of retinoic acid signaling called DEAB. Using single-cell transcriptomics, we were able to determine that these excitatory neuron lineages, the excitatory neurons that emerge during this differentiation are quite strikingly different from one another. And in particular, the presence of vitamin A in the media includes this group of excitatory neurons that are quite distinct from neurons that emerge when vitamin A was not added or when the inhibitor was added. And where we looked more closely at the expression of some of the known marker genes, including CUPTF2 or NR2F2, which is a marker of visual cortex occipital lineage, this marker was strongly enriched in both cases when vitamin A was withdrawn from the solution or when the inhibitor was added. In contrast, markers of prefrontal cortex identity that included actually co-expression of SADB2 and TBR1 in deep cortical layer neurons were both very strongly enriched in neurons differentiated in the presence of vitamin A. Moreover, we used immunostaining to independently validate these results. And when you stain the primary human tissue samples, and especially the subplate layer, with markers of prefrontal cortex, subplate neurons, and CUPTF1 in this case, you can see very clear distinction between those neuronal identities. And performing the same immunostaining experiment in cerebral organoids cultured in the presence of vitamin A strongly enriched or promoted the differentiation of excitatory neurons that express OTS2. In contrast, withdrawal of vitamin A promoted the differentiation of excitatory neurons that expressed strongly NR2F1, but did not strongly express OTS2. We also stained these both primary tissue and cerebral organoids with CTIP2, also known as BCL11B, and SADB2, which, as you can see in primary tissue, are co-expressed in prefrontal cortex, but differentially expressed in the visual cortex. And in cerebral organoids, we essentially found the same result. So to summarize my talk, I told you how single-cell genomics offers really an unprecedented and novel insights into human brain development. Using transcriptomics, we were able to distinguish these excitatory neurons that develop into quite substantially different identities, which we propose hallmark these proto-regions in the cerebral cortex. And very limited transcriptional differences seem to be found among progenitor cells. In contrast, the epigenomic or chromatin state changes that differentiate those progenitor cells seem to be much more pronounced. And so we propose a model that the chromatin state regulation in the cortical protomap may foreshadow the emergence of excitatory neuron lineages in different areas, proto-regions of the cerebral cortex. With that, I would like to end and acknowledge the people who did the work, and that included in my lab Ryan Zifra, a very talented graduate student, and Chang Kim, who is a bioinformatics specialist. I would also like to acknowledge the funding, and especially this project was supported by generous support from Psych&Code and the Brain Initiative, and I'm happy to take questions.
Thank you very much, Tom. That's a fantastic presentation. We're really pleased to have a question from the audience. Linda, I understand that you have one. If I can ask you to the chair. Yep. Thank you. Nice to see you.
Hi everybody. Fantastic talk, Thomas. I was thinking about the gradients of expression of EMX2, PAX6, etc. that you were talking about, and this idea that how do you go from the gradient to very defined regions? And one idea that you're putting forward, I guess, is the chromatin accessibility might be different between different cortical areas. And I wondered if you could bring that to link together with the gradient, because the amount of expression of these genes purportedly is what also drives the the functional area differentiation. And so I wondered if, for example, you could test that by overexpressing EMX2 in PFC cells and seeing whether you can drive chromatin accessibility to look like visual cortex, for example, or something like that. Do you want to make a comment on that?
Yeah, of course. I think that's a fantastic idea. And really, the technologies that enable that, I think, are only just really emerging, especially with the ability to implement, for example, CRISPR perturbations in primary or in iPS-derived cells. Of course, with those CRISPR perturbations, it is still really difficult to get very accurate perturbations or very accurate levels of transcription factor expression. I trained with David Price, who worked on PAX6 for a really long time. And the challenge with PAX6 is that it's a very, very difficult gene. So is actually retinoic acid signaling, because both not enough PAX6 and too much PAX6 results in the same thing because of the auto-regulation. And retinoic acid seems to be actually following the same rule, such that in order to get a proper specification, you really need to very carefully control the levels of these transcription factors. So I'm really interested in sort of testing this further functionally. Sort of the retinoic acid signaling pathway was something that stood out at us, because in addition to the differential enrichments of the transcription factors that you would have expected, including to CUPTF1 and SP8 in the prefrontal cortex and so on, we also found something that was very counterintuitive, because the retinoic acid tends to be thought of as a catalyzing factor, but in fact, we're finding it to be promoting prefrontal cortex. And because it's such a druggable target, we went after that one. But I think longitudinally, another really interesting type of experiment that we'd love to be able to do is to actually footprint the transcription factor bindings in different areas of the cerebral cortex, and to really understand, we can see enrichment in the chromatin loci that are accessible. Does it mean that the transcription factor is really binding there? And where, if we sort of go across the rostral axis, you know, do we see sharp drop off where the transcription factor is no longer binding? I think those are the types of experiments that we really need to start thinking of doing in order to really decode the logic of transcription factors.
Yeah,
so,
I think it's this levels hypothesis that you know, the whole field, it'd be great if we, you know, devoted a little bit more energy into trying to understand how different levels of transcription factors or morphogens even. I mean, you could be looking at FGF8 here as well, or other FGFs, FGF17, and to just see how that is, you know, read out essentially.
absolutely.
So, yeah, fantastic
That's a
talk.
great,
Thank you.
Thank you.
Thank you so much.
great question. Thank you,
Yeah,
Linda. Yes, of course. That's Song Hai, is that correct? Yeah, please come in.
It's a great talk. So I have a quick question. So you look at the one time point or multiple time point comparison between PFC and V1.
Mm-hmm.
How does temporal neurogenesis time difference that may contribute to the, that's the other point I try to see. So from your differentiation, epigenetic regulation, do you think that's influenced by much of the activity of autonomous input or things that may target those two regions? Because I was reminded from your talk that the recent study by Sunny Hsieh's group suggesting that activity does not contribute to much of the epigenetic regulation of the cortical. So I try to see how you think about those issues.
That's a fantastic question. Those are really great question. So to answer your first question, which was about how consistently can we detect the difference between prefrontal cortex and visual cortex? At the very least, I know this is an extremely shallow look. We've picked very divergent, you know, extremes of the gradient, and it has been recognized for many years that the prefrontal cortex and visual cortex mature at different rates. With single cell transcriptomics, we were able to actually profile at this point 20 individuals at different stages of development, ranging from approximately gestational week 13, so that mostly relates to deep layer neurogenesis. For anybody who works in mouse, that sort of approximate embryonic day 12, 13-ish, maybe more 12 than 13-ish, all the way to gestational week 22. And that the divergence occurs throughout that process. Now, there is a parallel study that was published by Basil Katasic et al. Institute comparing the mouse ALM, anterior lateral motor cortex, to the visual cortex, and they report the exact same pattern. And so we think that at least transcriptomically, those lineages, sort of the divergence is consistent. With epigenomics, because of the sort of cost of these experiments, we've only really profiled mid-gestational cortex, so around gestational week 20, but we profiled 3 biologically independent individuals. So at least at that stage, we think that this is a consistent pattern. So that's the first question. Your second question was about activity, which is I think, and one of the really interesting things that differentiates human and mouse is that the thalamocortical growth occurs actually much earlier than in the mouse. And so whether the presence of thalamocortical axons could be influencing the regional specification of the neuroepithelium has not really been extensively explored. There's beautiful work done by Colette DeHay's lab showing that when you have thalamocortical axons present in the neuroepithelium, the factors that are released by those include actually, especially FGF and factors that promote proliferation. A lot of mitogens are present. In the absence of thalamic axons, as shown by John Rubinstein's GBX2 knockout mice, at least at the level of some of the transcription differences across areas, those are still there. So even if you don't have thalamic axons, it seems like the differences emerge. Now, I don't know how much activity there is in the prenatal brain, which is highly hypoxic environment, and it's been well established that hypoxia tend to sort of dampen activity. So I think we just don't know enough about that process to say, you know, that is a strong contributing factor. But I completely agree with you. We know very little about those processes, and it would be wonderful to explore that.
Can I just make a point? Thank you for your question, Shanghai.
Thank you.
Tom,